Assalamu alaikum everyone. This is Sheikh Mueid, your biology A-level instructor from Elevare. Today we'll be looking at the question number 7 of the January 2025 Unit 1 biology paper. This was suggested by a student. So let's have a look. Before we begin, let's see some magic. Let's go. This will put less strain on your eyes. Okay. It says the structure of the cell membrane has been modeled to interpret the data available at the time. The diagram shows the timeline of 6 models proposed for the structure of the cell membrane. Okay, so we don't have a lot to do with this information. Let's just get straight to the question. They say Overton suggested that some types of solute can cross the lipid membrane, whereas other types of solute could not. Yeah, that's logical. Explain the difference between these two types of solute and why only one type can cross the lipid membrane. Before we get into the question, let me quickly draw out the basic structure of the cell membrane. We have two layers of phospholipid molecules with their tails in between. You should already know this much. These heads, or the phosphate heads as we call it, are hydrophilic, which means they have an affinity towards water. And these tails are hydrophobic. So they basically run away from any aqueous environment. Similarly, these tails are non-polar in nature, which means they will not allow a polar solute to pass through. That's basically because a polar solute will not be able to bind with the non-polar lipid tails. Hence, it will not be able to pass through the membrane. So the question asks us to explain the difference between these two types of solute and why only one can cross the membrane. As I already said, non-polar solutes can cross the lipid membrane. This is because the lipids in the lipid membrane are non-polar. So let's go ahead and mention that. Okay. Now we need to explain why only one type can cross. And that's because polar solutes cannot interact with the non-polar lipids in the membrane, obviously. So they cannot pass. A pro tip, always see what the question is asking for. This question had two parts. At first, it asked for the difference between these two types of solute and why only one could pass. So we said why only one could pass, and then we also said which solute can pass. Okay, on to the next one. I'll not try to pronounce that. This model was a single layer of phospholipids. Explain why the phospholipids in a cell membrane cannot be arranged in a single layer. Okay, let's get back to the structure I drew. If we look here, this entire layer and this entire layer is hydrophilic. So it will go towards the water or the aqueous environment. But the middle layers that we have here are hydrophobic. So they will always try to run away from the water or aqueous environments. If we have a cell and we have to have a membrane, the inside and the outside both will have water. So even if we try to keep one single layer of phospholipids, it will not stay like that. It will turn into a circle with the heads on the outside and the tails on the inside. That's basically how a phospholipid works. It will always try to shield the tails inside and keep the heads on the outside. So let's write it down. Phospholipids, we'll start with the structure. Have a hydrophobic tail and a hydrophilic head, as we mentioned already. If you're doing the paper with me, uh, then yeah, let's write it together. The tails will always orientate. You could also write position. This is just being fancy. To be away from aqueous environments on each side of the membrane. This is important because if we only had an aqueous environment on one side, let's say we had an aqueous environment only on this side, then we could still have a single layer structure, right? Because at that point, a single layer structure would keep the hydrophobic ends on one side and the hydrophilic ends on one side. But we don't have aqueous environments only on one side, we have it on both. So that's why we need a double layered structure. We'll finally say a bilayer. Bilayer is basically two layers. Is an appropriate arrangement. Yeah, done. Gorta and Grendel suggested that there were various types of phospholipids, but they were all arranged in a bilayer. Give two ways in which phospholipids can be different from each other. Let's look at the structure of a single phospholipid molecule. This is a phosphate group, and this is um, the lipid end, or as we call it, these are the lipid tail. The lipid tail are hydrocarbon chains of fatty acids, right? So they contain carbon and hydrogen. And the phosphate head is just a phosphate head. So when you have a hydrocarbon chain, you could have uh, saturated or unsaturated hydrocarbon chains, right? You could have different lengths of hydrocarbon chains. You could have, um, you could have different groups attached to the hydrocarbon chain. Let's just write two of them as that. The length of the hydrocarbon chain can be different. Perfect. On to the next one. These two models were the first. Oh no, this model was the first to include proteins. Explain why some membrane proteins would not be able to carry out their transport functions if they were located only on the surface of the phospholipid bilayer. Okay, membrane proteins, as shown here, this person said that these are only on the outside. Membrane proteins are basically needed for active transport and for facilitated diffusion. They act as channels. So, if this is one end of the cell membrane and this is the other, the proteins will span across it and allow substances to move directly into or out of the cell through its channel. It the presence of the protein will prevent the need for the solid to be non-polar because the solid does not have to interact with the lipids in that case. Let's write it down. Um, we'll write channel proteins and protein pumps. As we said, these need to span across the membrane For what? To transport substances. Okay. Now let's write what they do or what their transport function is. Channel proteins are required for facilitated diffusion. This is because this type of diffusion requires a channel protein for the movement of substances. And as we mentioned, protein pumps, these are required for active transport. These two are just two types of transport processes. Yeah. Active transport is where you need a protein pump and energy to transport substances against the concentration gradient. Now, this person implied that there was no space and that the thickness could be measured. Okay. Photograph shows two adjacent cell membranes. Thickness is 22.8 nanometers. Okay. Calculate the magnification. Okay. So since I'm on an iPad, I cannot measure the length here at the moment. So what you'll do is you'll have to measure this length using a ruler and then um, write it down somewhere. Let's just assume that it is 10 nanometers. Okay. And then they've given us that xy is equal to 23.8 nanometers. Oh, sorry, my bad. You'll measure in millimeters. You write it down as 10 millimeters, and the one they've given is 20.8 nanometers. Now we know the formula for finding magnification, which is image length divided by actual length. You could remember it like this, I by A. Now image length is 10 millimeters and actual length is 22.8 nanometers. We cannot find magnification if these units are different. So let's bring them down to a millimeter. Okay. And let's put that in our calculators. And we get 438,596 times because magnification is given in times. This is entirely because I guess this length, you'll have to measure it yourself and then write the correct answer. This is not the correct answer. I just use this length to show you how to do it. Okay, on to the next one. This is the last part of the question. Mm. In 1972, Singer and Nicholson proposed the fluid mosaic model of the cell membrane. Explain why their model has been given this name. Okay. This is the model that we still learn, the fluid mosaic model, this one. And if you have a look here, you'll see that there are things that are floating around and there are things that pierce through. Okay. And that's basically why the fluid and the mosaic parts in the names are given. The fluid part is because the phospholipids and the proteins can move around in the bilayer, hence the name fluid. Okay. And then mosaic. Normally, mosaic is used in um, floor design, styles, etc. Here it's also the same thing. You have proteins embedded at different places in the membrane, so it looks like a mosaic structure. The mosaic because the proteins are embedded throughout the membrane. Yes. For 2 marks, we've said why this name was given. And that concludes this question. Let me show you all the answers. If you missed any, take a screenshot and copy it down. Okay. This one. Okay, this structure is not needed for your answer. This is basically for me explain it. Yeah, yeah. This is just for examples purposes. You measure the correct length and then do the calculation. And here's the last answer. If you have any questions, make sure to leave a comment or ask us in our page. I'll be happy to help. And take care. Thank you for watching. Subscribe to our channel. And we hope that we can continue making such videos which will help you out.